Our next speaker is Euan Brennan from across town from me at the Institute of Genetics and Cancer at the University of Edinburgh. And Euan is very interested in stretch and cholangiocarcinoma.
Hi everyone, thank you. My name's Euan. I'm a second year PhD student in the Bolter Lab at the MRC Human Genetics Unit in Edinburgh. And my project is around exploring biomechanical signals within iCCA initiation. So as we're all aware, cholangiocarcinoma arises within the biliary tree, and the biliary tree is comprised of bile ducts, which are biological tubes that self-assemble to form this network. And as you've seen from some of Ed's work today, within the Bolter Lab, we use murine models to lineage trace cells that go on to form iCCA. And you can see in the image on the right there, those are the cells in red. And we're really interested in looking at those cells early within tumor initiation and trying to understand what processes drive neoplasia and dissemination from that tubular structure upon initiation. So using these murine models, we can begin to model tissue-level changes that occur not only in initiation, but throughout disease progression. So on the top panel there, you can see some 3D histology with the normal tubular structure on the left, with those cells in red, again, going to go on to form cancer. And interestingly, as we see disease progression and initiation through time, we see radial stretching of the cholangiocytes away from that ductular structure and sort of neoplastic lesions in replacement of what was normally a functional biological tube. And this is only exacerbated as disease progression goes on. And then on the bottom panel there, I just want to highlight when we do 2D histology, we can begin to morphologically phenocopy what we see in human patient disease. So as I said, I'm really interested in how biomechanical signals may be essential within iCCA initiation. And as you can see from those images, we move from this sort of representative diagram of a normal ductular epithelia, but through mutational acquisition and liver damage, we generate this sort of low-grade neoplasia, again characterized by stretching of the cholangiocytes away from the normal ductular architecture into the hepatocytes, and into the ECM, and we know at this time point there is also remodeling of this ECM, primarily through stiffening, and we propose that there are novel cell-to-cell mechanical forces as well as altered cellular and nuclear morphology. So one of the first things I did at the beginning of my PhD is try and set up a system by which we can introduce mechanical stress and begin to understand some of these biomechanical signals. So we used an organoid system derived from mice, both wild types and our double knockouts, the double knockouts having P10 and P53 loss. And when we treat these organoids with a drug called forskolin, we activate we open CFTR channels, which allows for ion influx. This generates an osmotic gradient, and we get reliable swelling of these organoids in a reasonably short space of time. So using this system, I've wanted to do two things. First, understand what are the early biomechanical responses to stretch, primarily through proteomics and phosphoproteomics using a reverse phase protein array, and I've performed this at 4 hours. And then to understand the transcriptional implications of these pathways, I performed bulk RNA sequencing 16 hours following stretch. And as I said, when we treat these drugs with forskolin, we can reliably stretch them. So you can both see sort of a nice visual representation of those on the left, and then just the quantification of that on the right. But interestingly, when we start to look at gross nuclear morphology and changes in nuclear morphology, we see two things occur in response to stretch. Firstly, an increase in the area of these nuclei, but this is also followed up by a reduction in their circularity. So the y-axis there representing 1 is a mathematically perfect circle, 0 being effectively a straight line. And you can see that reflected within the imagery on the left as well. So using the reverse phase protein array, as I said, we could reveal differential responses to mechanical stretch, specifically within our mutant cells. So I've just highlighted on the left some mechanical responders. So these are proteins that are involved in cytoskeletal remodeling, but also a known mechanotransductive pathway, YAP-TAZ. So we see activation of those, but interestingly, in response to stretch within our mutants, we see activation of mitogenic and growth factor-mediated pathways. And when we look into the transcriptomics, quite nicely we see that both the wild type and the mutant cells respond to stretch by differentially expressing genes. But interestingly, we see around 75% of genes are downregulated in response to stretch. However, I've highlighted on the heat map in red, there are clusters of differentially expressed genes that are specifically dysregulated in our mutant cohort. And as I said, we see activation of YAP-TAZ signaling within the reverse phase protein array. The next question is that being converted over into our transcriptomics data? So when we look at 16 hours at genes that are targets of YAP-TAZ signaling, we see that they're upregulated in response to stretch. And then when we perform gene set enrichment analysis and look at KEGG terms, we can see sort of quite a striking downregulation in a variety of metabolic processes, with the only upregulated terms referring to mismatch repair and DNA repair mechanisms. And sort of coming back to the changes that we saw in nuclear architecture at the beginning, I started to look at this complex called the link complex. And this serves to connect the nucleoskeleton through the nuclear envelope to the cytoskeleton through SUN and cache proteins. And when we look at gene expression for components of the link complex, we see that this is dysregulated specifically within our mutants in response to stretch. Interestingly, we also see a reduction in lamin A. And finally, as Ed quite handily introduced some of his single-cell data earlier today, I've started to have a look at some of the changes that we're seeing in our in vitro data set within his in vivo data set. And we can see that the changes that we see in expression in response to stretch are also differentially expressed within that data set when we look at mutant cholangiocytes versus our healthy cholangiocytes. So in summary, forskolin-induced stretch of our organoids reveals mechanical associated changes to both nuclear size and shape. Phosphoproteomics has identified early changes to mechanotransductive, growth factor, and mitogenic signaling pathways. When we look in our transcriptomic data, we see a near global downregulation of gene expression. However, within the mutant cholangiocytes, we see dysregulated expression of nuclear components in response to stretch, which we're beginning to investigate and phenocopy in our in vivo data sets. So firstly, I'd like to thank the AMMF and Helen for inviting me, and everyone in the Liver Growth and Cancer Lab and the Human Genetics Unit. Thank you.